Now let's take our second look in a bit more detail at the vertebrate immune system. We're doing it twice because it's a very important thing to get your head around, but it's reasonably complicated. Let's first take a look again at the structure at the level of the cell. In the bone marrow, so primarily in the large bones of our extremities, we have what are called pluripotent hematopoietic stem cells. That means that these are stem cells that can make cells that will go out into our lymph and our blood. And these stem cells can make many different kinds of cells, which are listed here. So as they go through their development, they can go into what's called a common lymphoid progenitor. And those will then develop either into B cells, T cells, natural killer cells, or dendritic cells. Or they can make a common myeloid progenitor that will go through a series of stages and become either a neutrophil, an eosinophil, a basophil, a precursor of a mast cell. If they make a megakaryocyte, the megakaryocyte will make platelets. And they can also make erythroblasts that then turn into erythrocytes. They then lose their nucleus and become a red blood cell. All of those things come out of the same stem cell in the bone marrow. And it's worth remembering that this would then be the origin of the different kinds of leukemias. Now, a bit about those cells. The B cells secrete antibodies, the T cells have a number of functions we'll go into, and the natural killer cells kill virus-infected cells before they can lyse. So the natural killer cells probably are actually the evolutionary ancestor of some of these other cells in the sense that they're part of the innate immune system. The myeloid cells, so they are things that then are going to be not primarily associated with the lymph system, but could be distributed throughout the body. They consist of macrophages. They actually eat bacteria, and they activate bacteria-killing mechanisms. The dendritic cells are the things that I've described as the guard cells, or the sentinels. They are out in the tissue, and they will take up antigen and present it in lymph nodes. So they will actually take up pieces of a pathogen and chop it up and present specific molecules in lymph nodes. The other cells are going to release signals from their granules when they're activated. Eosinophils are capable of killing a large antibody-coated multicellular parasite, so something like a digenetic trematode, schistosomiasis, or a hookworm, or something like that. And mast cells trigger inflammatory responses in tissues. So one of the things to remember about the immune system is that it consists of different cell populations, and those cells have different specific functions. The central action of the adaptive immune system is clonal selection, and clonal selection actually occurs in a lymph node. So you are to imagine that a dendritic cell has found a pathogen and has chopped it up into pieces and is ready to present a piece of it on its surface, and it comes into a lymph node. In the lymph node, it presents that piece of pathogen to a T cell, and the T cell then presents it to a B cell, and the B cell undergoes clonal expansion, okay? So there is, in the dark zone of a lymph node, a big population of different kinds of B cells. And the one that can recognize this specific presented chunk of parasite or pathogen is recruited. It undergoes a big clonal expansion. Then that population of B cells is checked. And there is a filter step here. It is checked to see whether there are any B cells that are producing antigens that react with self. And that screening is going on in the spleen and in the thymus and in the lymph nodes. And the accuracy of that screening is critical, because if some of the B cells manage to get through that react with self, then you can have an autoimmune disease. So selection then takes place. This is natural selection of B cell clones for the ones that are producing the specific antigen that's been detected by the dendritic cell and presented to the T cell, which then presents it to the B cell, and there's kind of a handshake that goes on at each of these steps. It's a system of control for specificity. And then there is some switching that goes on, and some of the B cells multiply, and they are going to be the ones that produce lots of antigen that goes out in the blood, and which can then bind to the invading pathogen. And other B cells are set aside and become memory. So you should think of the lymph nodes as being places where natural selection and specificity of action are occurring. And that should also help you to understand why it is that when we get an infection, we feel swelling in our lymph nodes, because the thing that's making them swell is this rapid expansion of the particular B cells that are being recruited to deal with that specific invader. Now, the central idea of the adaptive immune system is clonal selection. And it can be summarized like this. Every lymphocyte has a specific receptor with a unique specificity. An interaction between a foreign molecule and that receptor binding that specific molecule will activate that lymphocyte, and a differentiated cell that's derived from that activated lymphocyte will have the same receptor. So that's the clonal selection hypothesis. That cell makes that specific receptor, and it retains that in its descendants. So the lymphocyte that has a receptor that's specific for a ubiquitous self molecule will be deleted. It will be killed. And that will happen early in the development of these cells. And so you don't see, if in a healthy individual, you do not see lymphocyte populations, B cell populations, T cell populations that are mature that interact with self. They've been screened out. So if we look at this more diagrammatically, we see that a single progenitor cell will undergo some somatic recombination process that makes lots of different kinds of cells, each of which has a different specificity. Then the ones that can react with self are destroyed. So this would be a self-antigen. Those guys are destroyed. Those guys are destroyed. We're left with a pool of what are called mature naive lymphocytes. They're just waiting there. They haven't been selected yet for a foreign pathogen, but they don't react with self. Then a foreign antigen is presented, and a particular one, this one is yellow in this case, a particular lymphocyte lineage recognizes that foreign antigen and it multiplies. And then you have the vast industrial scale production of antibody to that particular antigen. The guy that came up with this idea was Sir Frank McFarlane Burnett, and he got the Nobel Prize in 1960 for it, and he was an Australian. Now, the diversity that allows this to run, this whole system to run, is generated in mammals by somatic gene segment rearrangements. That's called somatic recombination. It's a bit misleading. So you are to imagine that we've got lots of immunoglobulin genes, and they come in many different flavors, and in each particular cell, there is a process whereby only a particular combination becomes joined. And those joined segments are then transcribed. So actually the DNA first gets rearranged so that only particular combinations are present in that cell lineage, different ones in another cell lineage. And then the expression of that generates a unique receptor on each lymphocyte. Okay, so you will have a population of cells that are sitting there in lymph nodes, and one cell might be presenting a receptor that would be effective against the common cold, and another cell might be expressing a receptor that would be effective against malaria. And since that person hasn't been infected yet, those cells are not yet active. They're just sitting there waiting for a chunk, a molecule that's specific to a particular pathogen to be presented to them. And they don't know which pathogen is coming along. They have all kinds of different receptors on their surfaces. There are so many different possible combinations because of the multiple gene families and the multiple alleles that completely new pathogens can be identified and can elicit a response. So, antibodies defend the host mainly in three ways. They neutralize it, they opsonize it, or they activate complement. An antibody is a protein that binds specifically to a substance, which is its antigen, and they are known collectively as immunoglobulins. Antibodies are produced by plasma cells in response to infection or to immunization. So either an infection or a vaccine will eventually result in the production of antibody. Opsonization is the alteration of the surface of a pathogen or some other particle so that a macrophage can eat it. And that can be done either with antibodies or it can be done with complement. And the complement system, which is part of the innate immune system, is a set of plasma proteins that attack extracellular pathogens. They coat it, and that makes it possible for them to be recognized and ingested by phagocytes. And some pathogens can be killed directly by complement. They smother it. So if we think about what antibodies do, some antibodies are going to interact with bacterial toxins, and they will neutralize them so that they can be ingested by macrophages without damaging the macrophage. Other antibodies interact with bacteria that are out in intracellular space. They coat it, so they bind specifically to surface molecules on the bacterium. And that can then be recognized by the macrophage as, oh, that is a particle that I ought to eat. It's coated with something that's specific to non-self, and it's eaten by macrophage. Another possibility is that there are bacteria out there just in the plasma rather than in extracellular space, and in the blood, they get coated by complement, and then they are lysed and ingested by macrophage. There's a whole another set of processes that occur when a cell's already been infected. So if you have a virus-infected cell, it will actually express the fact that it's infected. It often will take a piece of virus and present it on its surface. It will then be recognized by a cytotoxic T cell. So this is a T cell which has been recruited with a receptor on its surface that matches the particular piece of virus that's being presented on the surface of the cell. And that cytotoxic T cell will either signal that this cell should commit apoptosis, it should commit suicide, or it will kill it by actually producing enzymes that dissolve its cell membrane. And the whole point here is to try to kill the cell before the virus can take it over and reproduce and lyse and produce lots of baby viruses. There is a special kind of defense against intracellular infection. Now, there are lots of good reasons why a pathogen should want to hide inside a cell. Out there in the blood or in the intracellular space, it's in a pretty open environment where the immune system can attack it pretty easily. So some pathogens have actually evolved to live inside the cells of the host. A good example is a tuberculosis bacterium, and it actually lives inside macrophages. And that's pretty amazing because the macrophage is the predator that normally would be killing things, but the tuberculosis bacterium is good at infecting macrophages. And what the macrophage then does is it uses enzymes in its lysosome to chop up the bacterium and then present pieces of bacterium on its surface. So these are bacterial antigens presented on its surface. And a T helper cell will come along and it will activate, this will be a macrophage which has used its lysosome to become activated, and the T helper cell will recognize that and kill it. So we have intracellular machinery that basically allows us to respond with great specificity, and it's very interesting that infected cells essentially are set up to commit suicide. So where is it that antigen is recognized by antibodies, and where is it recognized by the major histocompatibility locus receptors? Let's run through a series of cases here. Case one is that there is a virus, and there's a virus protein which is in the cell. Okay, so the virus has infected the cell. It is synthesizing protein in the cell, so there's viral protein coming off the endoplasmic reticulum. The peptide fragments of the viral protein are bound by MHC class I molecules, okay, in the endoplasmic reticulum, and then these bound peptides, so these little chunks of viral protein, are transported and presented at the surface. And they are then recognized, and that cell is attacked by a cytotoxic T cell. Let's take a second case. So this is the bacterial case. The bacterium infects a macrophage, it enters a vesicle, and it's producing peptide fragments. The bacterial fragments are bound by MHC class II molecules in vesicles, and they are then transported to the surface and presented, and the same thing happens. So MHC class I binds with viruses, MHC class II binds with chunks of bacterium. Third case, a B cell is out there, and it's got an antibody on its surface, and the antibody binds to a specific antigen. So a chunk of pathogen is floating around in the blood, and a B cell recognizes it. That antigen is then internalized. It is degraded into peptide fragments. Those fragments bind to an MHC class II molecule, and the function of that is to take it to the surface and present it. Okay? Now, at that point, a number of things can happen. This could lead to the multiplication of that B cell clone and to the production of a lot more antigen, therefore, and it probably will also, it can lead to the death of that B cell if a T cell tells it to kill itself. Now, there are three kinds of T cells that are triggered when a peptide that's bound to an MHC receptor is recognized. So you've seen these MHC receptors that are working intracellularly and taking fragments and moving them up to the surface of the cell. The T cells can kill, so the cytotoxic T cell can recognize a complex of viral peptide on the surface and kill a cell infected with a virus, or they can activate macrophage and B cells. So they can recognize a complex of bacterial peptide that's got MHC class II, it will activate a macrophage, or a helper T cell will recognize a complex of antigen peptide, and it will activate a B cell. And then the activated B cells proliferate and they produce antibody. This may all seem very complicated, but in fact, what's going on here basically is that a system has been set up that's very specific and that has safeguards built into it. Because remember that all of these cells, these cytotoxic T cells in particular, and the B cells that can produce a tremendous amount of antigen, are extremely dangerous if they make the mistake of recognizing or of misrecognizing self as being foreign. And so each one of these is like the situation with two officers sitting by an ICBM, and each of them has to turn a key in order to activate the ICBM. There have to be handshakes all the way through this signaling cascade to make sure that you don't activate something that will kill self, at the same time very effectively activating large amounts of stuff that will kill invaders. So to summarize, Adaptive immunity greatly increases the range of protection against pathogens, and it adds memory. So it can deal with things that have never existed before, that are newly evolved, because it's producing such a huge array of possible combinations. It operates on the principle of natural selection. There is somatic generation of antibody diversity, one antibody per clone, and then there is selection of clones that match the pathogen antigens. The adaptive immune system uses different mechanisms to deal with viruses, intracellular pathogens, and extracellular pathogens, and has differentiated different sorts of cells to specialize on each of those functions.